All right, folks, welcome back. We're going to launch into the final third of our symposium with a talk by Sylvia Jacobson and Kyle Derby from Chesapeake Bay, Maryland.
All right, thanks. So today we'll be presenting on how we're using the long-term wetland and water level data to investigate how microbial communities in Chesapeake Bay, Maryland wetlands may signal vegetation change. And this is part of an ongoing project, part of my PhD work as a Margaret A. Davidson Fellow with the Reserve. So in the Chesapeake Bay, Maryland, we can already see that some of our wetlands are changing very rapidly under sea level rise. For an example, this is a picture of Menai Bay on the eastern shore of Maryland, where you can see dead trees standing that are killed by increased flooding and salt pulses under sea level rise. And you can see some of the wetland vegetation, the Spartina patens and the Phragmites moving into the upland. But it's not as well known how sea level rise is impacting the soil biochemistry. So the wetland organic rich soil contains a mixture of up to thousands of different kinds of carbon molecules and is a habitat for a diverse array of different microbes that break down that carbon and use it for energy. So how are these carbon molecules and microorganisms changing under sea level rise? And how might that affect the wetland stability and ability to build vertically?
Thanks, Sylvia. And so one of the ways that we were able to support a project like this here at our reserve was looking back at our long-term data that is part of our WWL monitoring. So we've been doing vegetation surveys from 2008 until now at all three of our components. Uh, we're using our longer running transects for Sylvia's projects. So that's 6 at Menai, 14 at Jug Bay, and 15 at Otter Point Creek. We also have been reading SETs at all of our components for about the same amount of time. There's a little bit of, wiggle room there at the start date. But again, there's 17 SETs at Jug Bay and 12 at Menai Bay. And then we're also combining that with our water quality monitoring for Sylvia's projects. So this project that Sylvia is performing really shows how the WWL data can be used for something completely different than just looking at what we've been looking at before. This has really allowed Sylvia to kind of go back in time almost, look at where we've started in our wetlands and where we are now, and then compare the microbial data that we have now, and then maybe even be able to predict the future.
Okay, thanks. Yeah, so exactly. So the goal is to be able to use all this long-term data from the vegetation surveys, the SET, and the water quality, and be able to connect that to the present day biochemistry, the microbes and the carbon molecules. So first, across the three reserve sites, we reviewed these long-term data sets to select sites for this biochemistry analysis. Sites that have either changed over time or remained stable. So here is the salinity data for these sites over time, and we found that there was no change in either average salinity or the number of high salinity pulses over time across all of these reserve sites. So these graphs note that they're different scales on the y-axis here, since Jug Bay and Otter Point Creek are tidal freshwater marshes. And some of these salt pulses can be due to storm events, but also things like road salt application. But since we didn't see any trends in change over time at these sites, we focused on changes in flooding due to changes in elevation and vegetation changes to connect to the biochemistry. So, for example, at Jug Bay, a riverine tidal freshwater marsh, using the wetlands and water level data, we found some sites that lost elevation over time and show a loss in plant cover and a shift toward a lower marsh plant community. So these graphs are showing plant cover on the y-axis. And even without, and different colors correspond to different species, but even without understanding which colors, which species, you can generally see that for these sites, the cover is decreasing over time and there's a change in plant community composition. And then also at Jug Bay, we found other sites that showed increases in elevation, approximately keeping up with sea level rise, that had more stable plant communities or even a shift toward higher marsh plant communities. And these are some photographs of an example of what some of these sites look like. A site on the left where the plant community has remained relatively stable over time, and a site on the right that lost elevation and has seen a shift, loss of plant cover, change in plant species over time. So based on this analysis of the wetlands and water level data, we're able to choose sites that either had a declining or a stable plant community. And we collected over 250 soil samples from these different sites. And then we're sequencing the soil microbes, and we're also conducting carbon chemistry analyses where basically we're doing a high resolution mass spectrometry technique to look at the different kinds of carbon molecules. And we expect to see differences in microbial communities and carbon chemistry at sites that have declining plant communities versus those that are more stable. So the reason we're interested in this and the hypothesis is that potentially these microbes and the carbon chemistry can act as as tracers of these past changes in the wetland. So, for example, just by looking at the specific microbes and carbon molecules at a site, you might be able to understand the history of the site in terms of how the flooding and plant community has changed over time. That way, in the absence of over a decade of monitoring data, you can learn a lot about a site's history just by collecting some surface soil samples and doing some DNA sequencing and carbon chemistry analyses. Also, comparing the soil microbes in sites that have changed versus sites that haven't changed lets us assess the impact of sea level rise on soil biochemistry using a time for space substitution. So although we haven't actually monitored how the soil biochemistry is changing over the last 12 years, by by comparing sites that have experienced these different changes, we can still get a sense of how the changes in soil biochemistry is happening over time and how changes in flooding and vegetation are affecting that. So as a PhD student, it wouldn't be possible for me to monitor long-term changes in marsh vegetation and elevation changes for over a decade. But using wetland and water level data, I'm able to have this understanding of the site history to be able to connect these measurements of soil biochemistry to the broader changes on a landscape scale across these sites. So the project is still ongoing, so I don't have the biogeochemical data to share, but that's our study plan and what we've been doing so far.
That's great. Great example of the MAD fellowship linking into wetlands and water levels. There was a question in the chat from Matt of, are you able to distinguish carbon isotopes in those soil samples?
So you could look at carbon isotopes. We're not focusing on carbon isotopes. Instead, what we're looking at is the carbon molecular composition. So like how many in each carbon molecule, how many carbons, hydrogens, oxygens are there using mass spectrometry. So there are different kinds of ways you could fingerprint carbon, but that's the way that's what we're doing.
I have a question about your lack of salinity changes. That's in the sort of water column salinity, but what the plants have their roots in could be affected by groundwater and saltwater intrusion. Would it be worth looking at that too?
Yes. So yes, you're right. This is just That's just from the main water body where the water quality was monitored. So it's not, there could be changes in the port water that's not reflected in that. So that's something to look into. But yes.
Okay, well, thank you so much.